Okay, now I'll take you through a simple 3 fluorescence channel capture and overlay. So to do this, what we're going to do is we've got our slide mounted on the stage here. We have the objectives below the stage that can be selected by using this wheel. And as I switch between positions, it updates and tells me which objective is in which position here. We're going to be looking at some cells that are stained in 3 fluorescence channels, GFP, Texas Red, and DAPI. So to do this, we're going to start off with the GFP channel, and I just click on the GFP button here. The motor takes the light cube to the GFP position, and we're now looking at GFP live. And I can move my sample here with the stage controls and select an area of interest. If I find something that looks particularly interesting, I can zoom in on it, or I can increase and decrease the fluorescence intensity by moving this slider up and down here. If I want to look at it in more detail, I can move this zoom bar up here, and we can make fine adjustments to the focal plane and then click capture, and we'll have that entire GFP channel. In order to capture the other channels, I can do the same sort of thing in sequence and it will overlay, or I can actually sit here and ask it to collect the remaining 2 channels by selecting them in the software here and clicking capture, and it will go ahead and add those 2 to the image. Once we're in here, it's possible to go in and zoom in, look at an area in detail. We also have the capability of turning on and off a scale bar, moving this around on the screen, selecting a location for it, or we can put on a grid, select a density of grid here, say every 100 microns, so you can tell how many cells there are per unit area in your image. And when you save the images will be saved with these overlays. We also have the capability of showing a histogram of the intensity distribution here for the channel. There's the capability of going in and doing annotations, so you can select a tool here and measure an area within a rectangle. Gives you calibrated area here. This is because the calibration of the system is automatically detected and known based off of the objective that it's using. Some of the other tools that are in here, we have the capability of going in and doing cell counting. So if we do that, we can bring this tool up here, and it automatically isolates the DAPI channel. We can go in and select this area here, showing us some representative nuclei that have been stained with DAPI. And then I usually try to switch over to the monochrome channel, so it looks a little easier to find the cells. And then we select a target, so we just kind of identify what our targets are here. This is a target, this is an example of a target, and so is this one. And then we select a background, and by selecting the background, it will automatically go in and calculate the cells and circle them for us here. There's the ability to go in and make further refinements to this. We can actually go in here and change this based off of the area, and then take out the smallest units here, which are probably not nuclei but fragments of cells, and then get a revised cell count here, or object count of 64 cells. When we first turn this on, you can see that we're not in focus here. So I can either use the coarse focus and the fine focus here. I can use the mouse wheel to focus this, but we also have an option in here to do autofocus. So if I click on the coarse autofocus, it's going to go through this range that's defined here, and it will try to find approximately the best location. It's actually picked this one, and if I double click, I'll get a zoom, and it's actually done a really nice job with the autofocus there. We don't need to refine it any further. If I want to, I can move my mouse wheel up and down here, but you can see it's already looking pretty good. The M5000 can also do Z-stacks. So in here, we can show that by going into the Automate tab, selecting Z-stack, and we get this set of options here. We can go in and have this set above and below our location that we want for the focal plane. And then once we've entered those values for the top and bottom, it will automatically compute the number of images that we're going to get, and it will execute that when we click on Start. It's going to go through and capture 15 planes of focus here. And at the end, it opens up a folder where it's put those images, and we have the ability to scroll through the images. Right here, if we click on the results, this is midway through the stack, and we can just bring this up and down. We can actually just zoom this up a little bit. And you can see as I'm focusing up and down through this sample, we're going above and below that optimal focal plane. We also have a review tab where you can go in and look at images that you previously acquired on the instrument. These images can come from the local hard drive built into the microscope, or from a flash drive that's attached, or they can be retrieved from the cloud. In this case, we're looking at some images that are on a flash drive, and when I click on these and look at them, they load up and they allow me to go in and zoom, pan, look at various details, and review my images. The illumination system is still based on an LED light cube, which has the LED built inside with electronic controls. It can be turned on and off as many times as you like without shortening the lifespan, and the lifespan is over 50,000 hours. With 50,000 hours of use, you can actually turn it on for 8 hours a day, 7 days a week, and it will be over 17 years before you hit 50,000 hours. Another feature of the M5000 is the ability for it to take onstage incubation. With an optional onstage incubator you can attach to the microscope, you can keep your cells alive for extended periods of time and do time-lapse imaging. With the onstage incubator, we can control a variety of features. We can go in here and show that we've got access to temperature and humidity, as well as the gas combinations here. The incubator can actually mix up to 3 different gas lines, so typically CO2, oxygen, and if you want to study hypoxia, we can put in nitrogen, and you can have control over that by specifying different stages of incubation. So under time-lapse, we can go in here and set up our channels. We can use the incubator and turn on autofocus. We can go in and select transmitted for the autofocus channel, and then we can define different runs. So the first run could be a baseline where you're just looking at the cells in normal conditions. We can set that up for a certain duration, say 10 minutes. Then we can go to the next phase where we can switch over and save this as a TIFF or a PNG file, in addition to creating a movie file at the very end, so that you can review the entire sequence. We can put in a run after that baseline, take our cells into hypoxic conditions and study the effects on them. This is all something that can be added to the M5000 very easily, and it's completely integrated and controlled through the existing software. There's no additional charge for the software component. The EVOS M5000 includes access to a digital Smart Start online guide, which you can log into. It has various information. It has a system overview that gives you the different components in here. You can also look at instrument care. There's information on how to change the light cubes or how to change an objective. And all this consists of animations. So when you click on this, it highlights the tools and parts that you'll need. This is a spare objective that we'll be adding to it, and it takes you through step by step. So basically you go in and you have to remove the slide holder or the cover that's going over the objectives. Then we're going to move the stage so that we have access to the objective turret underneath here. Then it has information about removing by unscrewing. You can see it's animated. We unscrewed and removed the objective, etc. So it takes you through step by step the things that you need to do to put in a new objective and configure it in the software. Another capability we've added to the M5000 is the ability to synthesize color images for you. Now, the instrument normally comes, and does come in this case, with a high sensitivity monochrome camera for optimum fluorescence detection. However, our engineers have put together a beautiful system that allows you to strobe red, green, and blue LEDs and synchronize that with the camera capture and synthesize a color image. So I'll give you an example of that. We've got a slide here, a tissue slide, stain of cirrhosis of the liver, and I'm going to go over here to the RGB transmitted light mode and turn this on. And you can see that the monochrome camera is giving us a beautiful rendition of color here of the tissue. And if you look closely at the illumination here, I can put my hand down here, you can see that there's a flickering, and that is the red, green, and blue LEDs being strobed over the sample to give us the color information. With this kind of capability, you're able to go in and look at standard histology slides, anything that has any kind of color information, and capture beautiful images with it. Anybody can walk up to the microscope and get started on it with a minimum of training.